Welcome to a new deep dive. Today, we are going right inside your body's amazing communication network.
Yeah, forget your phone signal. This is the real deal. Biological.
Exactly. Nerve signals, everything your body does. I mean, everything from thinking to breathing to digesting. It all relies on these signals traveling down specific routes using very precise chemical messengers.
It's incredibly complex. And, most of it just happens automatically. You don't even think about it.
That's what's wild. And today, we're really going to unpack the basic science. How do those signals actually jump from one nerve cell to the next, or from a nerve to a muscle?
Right. Well, look at the system you do control, deciding to, say, weigh your hand. That's the somatic system.
Mm-hmm.
And then the much bigger part, you don't directly control heart rate, digestion, all that autonomic stuff.
And we're digging into some really detailed source material here, looking at the nitty-gritty of neurotransmission.
Yeah, the core science.
Our mission, really, is to get a handle on how this nerve communication actually works. Who are the players? The nerves, the chemicals, the receptors, the receiving stations.
And crucially, why does this matter? Because understanding this is key to, everything your body does, and also how a lot of medicines work.
We're hoping to make this complex stuff feel, surprisingly clear, maybe even kind of cool.
Let's give it a shot.
Okay, so let's start big picture. Let's map out this network. We're talking peripheral nervous system connecting the brain and spinal cord out to, everywhere else.
Mm-hmm. And there are two main output highways we're focusing on today.
Right. First up, the somatic nervous system.
Yep. Your voluntary control center. Allows you to, walk, talk, type. It sends signals straight from the spinal cord out to your skeletal muscles, the ones you choose to move.
Simple enough. Then there's the other one, the autonomic nervous system, the ANS.
The unsung hero.
Yeah.
Working 24/7 behind the scenes.
Totally. Handles all the involuntary stuff. Your heart speeding up or slowing down, your stomach digesting, breathing, glands, like sweating or making saliva.
It's sometimes called the visceral system, or involuntary. And its reach is just huge. It talks to pretty much every internal organ, smooth muscles, glands.
And there are some really basic differences in how these two outgoing systems, autonomic and somatic, are set up. Right?
Big differences, yeah. Think about where they go. Somatic nerves, only skeletal muscle.
Okay.
Autonomic nerves.
Yeah.
Everywhere, but skeletal muscle.
Yeah.
Heart, gut, blood vessels, you name it.
And the connections, they're different too. You mentioned these relay stations earlier.
Exactly. That's a key difference.
Yeah.
Somatic nerves are like a direct line.
Yeah.
Spinal cord straight to muscle fiber. One connection.
Okay.
Autonomic nerves often have this extra step, a pit stop. It's called a peripheral ganglion, basically a cluster of nerve cell bodies outside the brain and spinal cord.
So the signal goes from the spinal cord, stops at this ganglion, and then another nerve takes it the rest of the way to the organ.
Precisely. That ganglion synapse is critical. Somatic motor nerves. They only synapse inside the central nervous system. Autonomic nerves have that extra peripheral synapse.
And structurally, they look different, too. The autonomic system forming networks.
Yeah, these widespread webs or plexuses across organs. Somatic nerves are more like dedicated cables. And the autonomic fibers after that ganglion, they're usually slower. They lack that fatty insulation, the myelin sheath.
Myelin speeds things up.
Right. Which makes sense for somatic nerves. You need quick commands for muscle movement. Autonomic control can often be a bit slower, more gradual.
And that difference in speed and structure, it leads to a really different outcome if the nerve gets damaged, doesn't it?
Absolutely. Cut an autonomic nerve supply. The target organ, like smooth muscle or a gland, it often keeps some level of activity. It might not work perfectly, but it's not dead in the water.
But if you cut a somatic nerve going to your bicep...
Paralyzed. That muscle can't contract without the signal. Complete loss of voluntary control.
Wow. Okay. Now, we're mostly talking outgoing signals, but information flows back too, right? From the organs to the brain.
Oh, constantly. Sensory feedback, visceral apparence, signals about pressure, stretch, chemicals, temperature, pain.
Yeah.
All coming back from your insides.
And where does that information go?
It travels back via various nerves, some cranial nerves, some spinal nerves, and feeds into the central nervous system. What's interesting is how signals from different organs often converge. They meet up in specific areas, like the solitary tract nucleus in the brainstem.
Convergence. So the brain gets a sort of integrated picture of what's happening internally.
Exactly. It helps coordinate responses. And this highlights that the brain doesn't always keep autonomic and somatic totally separate. There's overlap. You can have reflexes that are purely autonomic, triggered right at the spinal cord. Think sudden sweating or blood pressure jumps.
Without even involving the brain consciously.
Right. And higher up, you've got major integration centers, the medulla, parts of the hypothalamus.
Yeah.
They're crucial. The hypothalamus especially is like a master controller.
What does it control?
Oh, tons. Body temperature, metabolism, blood pressure, fluid balance. It even links autonomic responses to emotions. It gets input from all over and sends out commands to adjust things.
Okay, so let's zoom back out to those two main branches of the peripheral ANS again, the sympathetic and the parasympathetic.
The two divisions of the outgoing autonomic system, yeah.
Let's start with the sympathetic. This is the fight or flight one, right? Gets you ready for action.
That's the classic description. Its nerve fibers originate in the middle section of your spinal cord, the thoracic and upper lumbar segments, T1 to about L2 or L3.
And the structure, how do the signals get out?
The first nerve fiber leaving the spinal cord, the preganglionic fiber, is usually quite short.
Okay.
It travels to one of those sympathetic ganglia we mentioned. These ganglias are located relatively close to the spinal cord, either in chains running right alongside it, the paravertebral ganglia, or a bit further out in the abdomen, the prevertebral ganglia.
And then the signal gets passed on.
Right. From the ganglion, a long postganglionic fiber travels the rest of the way to the target organ, heart, lungs, blood vessels, sweat glands, wherever.
And I remember the source highlighted something about the connections in that ganglion. One incoming fiber can talk to many outgoing ones.
Yes, that's key. Significant divergence. One preganglionic fiber might synapse with, say, 20 or even more postganglionic neurons.
So a single alert signal from the spinal cord can spread out really wide.
Exactly. It allows for a diffuse body-wide activation, perfect for mobilizing everything quickly in an emergency.
Makes sense. And the chemical messenger used by most of these sympathetic postganglionic fibers is?
Norepinephrine, or AE. That's the main one. Though interestingly, there are exceptions. The sympathetic nerves going to sweat glands, they actually use acetylcholine, or AKey.
AOI, which is usually the other system's chemical, right? The parasympathetic system.
Exactly. The parasympathetic is generally seen as the rest and digest system, conserving energy, promoting housekeeping functions.
And where do its nerves originate?
Two main places. The brainstem, traveling out with cranial nerves, particularly a big one, the vagus nerve, cranial nerve X, and the very bottom of the spinal cord, the sacral region, S2, S4. So craniosacral outflow.
Okay, so top and bottom compared to the middle for sympathetic. And the structure is flipped too.
Pretty much flipped, yeah. Here, the preganglionic fibers, the ones leaving the brainstem or sacral cord, are very long.
Long? How long?
They travel almost all the way to the target organ.
Wow.
They only synapse in those terminal ganglia, which are located right on or even inside the wall of the organ itself, like in the heart wall or the gut wall.
Which means the postganglionic fibers must be
super short, just tiny connections within the organ.
So compared to the sympathetic system's wide broadcasting, this setup seems much more targeted, localized?
Generally, yes. It allows for more discrete control over individual organs. Though even here, especially in complex networks like the gut, one incoming fiber can still influence a lot of targets.
And the main chemical messenger for the parasympathetic system?
Primarily acetylcholine, ATH. It's the workhorse here, though some parasympathetic nerves use other things, like nitric oxide.
And you mentioned something earlier. The parasympathetic system is essential for life.
Yeah, the source notes that. While the sympathetic system is vital for handling stress, you can technically survive without it, albeit poorly, but completely losing parasympathetic function. Not compatible with life. You need it for basic functions like digestion, heart regulation, emptying your bladder.
Makes sense. Okay, and while we're near the gut, we have to quickly mention the enteric nervous system, the ENS.
Ah, yes, the second brain in your gut. It's this incredibly complex, semi-independent network of neurons within the walls of your digestive tract.
It gets signals from the sympathetic and parasympathetic systems, but it can also run the show locally.
Largely, yes. It controls muscle movement, secretions, blood flow, all within the gut. And it uses a whole cocktail of neurotransmitters. A-sheet, ATP, serotonin, peptides like substance P, nitric oxide. It's really sophisticated.
And that detail about the interstitial cells of Kala. The nerves don't always talk directly to the muscle.
Right. Often, the motor nerves in the gut synapse onto these specialized intermediary cells, the ICCs, which then pass the signal along to the smooth muscle cells. They act like pacemakers and signal transducers. Pretty neat.
So quick recap on the comparison. Sympathetic action, middle spine origin, short pre-long postganglionic fibers, ganglion near spine, diffuse effects, mostly NE.
Parasympathetic rest digest, brainstem sacral origin, long pre-short postganglionic fibers, ganglion near and target organ, localized effects, mostly Aishi.
And somatic motor voluntary, direct from spinal cord to skeletal muscle, single neuron, uses Aishi for fast activation.
You got it. And just a couple more structural points on those autonomic fibers.
Go on.
Those postganglionic fibers, they don't just have one single ending. They often have these little swellings along their length like beads on a string called varicosities.
Varicosities.
Yeah. And these are packed with the vesicles containing the neurotransmitter. So instead of releasing chemicals at just one point, the nerve can release them along its path, influencing a wider area of the target tissue.
Ah, like a sprinkler hose instead of a single nozzle?
Kind of like that, yeah. And in smooth muscle particularly, the cells themselves are often electrically connected by gap junctions, little protein bridges.
So the signal can spread directly from cell to cell.
Exactly. Helps the whole muscle sheet contract in a coordinated way.
Okay, structure makes sense. Now, the really cool part. How do these two systems, sympathetic and parasympathetic, actually play together at the organ level?
Often they have opposite effects. Functional antagonism. It's like having an accelerator and a brake for many bodily functions.
Like the heart.
Perfect example. Sympathetic stimulation speeds up your heart rate and makes it beat stronger.
Okay.
Parasympathetic stimulation via the vagus nerve slows it down.
Or the pupils.
Sympathetic dilates them, lets more light in. Good for seeing danger. Parasympathetic constricts them better for focusing up close, protects the retina in bright light.
And digestion.
Sympathetic generally inhibits digestion, slows down gut movement and secretions. Not a priority when you're running from a bear. Parasympathetic stimulates it, gets things moving, increases secretions for digesting food.
So they work in opposition to maintain balance or homeostasis.
A lot of the time, yes. It's this constant dynamic balance. But it's not always direct opposition. Sometimes their actions can be complementary, both contributing to an overall function, like in male sexual response, for instance. And remember, the sympathetic system isn't just for emergencies. It's active all the time, making fine adjustments to things like blood vessel tone based on your posture or activity level.
Right. It's not just all or nothing, fight or flight.
No. And when a major response is needed, the sympathetic nerves can fire together and get a big boost from adrenaline or epinephrine released from the adrenal glands right into the bloodstream. That's the sympathoadrenal system working as a unit.
While the parasympathetic side handles the essential baseline stuff, slowing the heart, aiding digestion and absorption, eliminating waste, the day-to-day running of the ship.
Precisely. Vital for normal life.
Okay, fantastic overview of the map and the main divisions. Now, let's zoom right in. All the way down to the synapse, that tiny gap. How does the message actually jump across?
This is where the chemical language really comes into play. The whole idea that nerves communicate chemically was a pretty revolutionary concept.
It wasn't always obvious.
Not at all.
Mm-hmm.
People noticed similarities like adrenal gland extracts mimicking sympathetic nerve stimulation, but proving it was tricky. The breakthrough really came from Otto Loewe in 1921, a classic experiment.
Oh, this is the frog heart story, right? I love this one.
It's brilliant. He had two frog hearts, kept beating in separate containers of fluid. He stimulated the vagus nerve going to the first heart.
And the vagus nerve is parasympathetic, so...
Slowed the first heart down. Exactly. Then, and this is the genius part, he took some of the fluid bathing that slowed down heart and transferred it to the second heart's container.
And?
The second heart, even with no nerve attached, also slowed down.
Wow. So something chemical must have been released from the first heart's nerve into the fluid.
Precisely. He'd shown chemical transmission. He called the substance vagus stuff. Vagus substance. We now know it was acetylcholine, a key.
Incredible. Just elegant.
Totally. He later found an accelerant stuff, too, something like adrenaline, when stimulating accelerator nerves. It was the first direct proof. Later work confirmed NE as the main sympathetic nerve transmitter and EPI, adrenaline, as the hormone from the adrenal gland.
So how do scientists today confirm something is a neurotransmitter? There are criteria.
Yeah, a checklist, basically. Is the chemical actually in the nerve terminal? Do enzymes needed to make it exist there? Is it released when the nerve fires? If you apply the chemical artificially, does it mimic the nerve's effect? Yeah. And do drugs that block the nerve's effect also block the chemical's effect, and vice versa?
Makes sense. Though you mentioned exceptions, like nitric oxide.
Right. NO is weird. It's not stored in vesicles. It's a gas made on demand when needed, and it just diffuses across membranes. Doesn't fit the classic model perfectly.
Huh. And nerves don't always just release one chemical, do they?
No, that's another layer. Co-transmission. Many neurons release a classical transmitter, like ACA or NE, plus one or more other signaling molecules, often peptides like NPY or substance P or maybe ATP.
Why do that? Why release multiple messengers?
It adds nuance, complexity. The different chemicals might act on different timescales or activate different receptors, allowing for much finer control over the postsynaptic cell's response.
Okay, so let's break down the step-by-step process. How does that signal actually get across the synapse? From electrical pulse to chemical message received?
Right. Step 1. The electrical signal, the action potential, zips down the axon, the nerve fiber.
That's the movement of ions, sodium and potassium, across the membrane.
Primarily, yes. Voltage-gated sodium channels open, letting positive charge rush in, depolarization. Then a potassium channel's open, letting positive charge rush out, repolarization. That wave travels down to the nerve terminal.
When it gets to the end, the terminal.
That electrical change triggers the opening of calcium channels. Voltage-gated calcium channels.
Okay, so the electricity hits the end, calcium channels open. What does the calcium do?
Calcium rushes into the nerve terminal, and that influx of calcium is the absolute key trigger for the next step, transmitter release.
How does calcium trigger release?
Inside the terminal, you have those tiny sacs, the synaptic vesicles, filled with neurotransmitter molecules. The incoming calcium ions bind to specific sensor proteins, like synaptotagmin, located on the vesicle membrane.
Okay.
This calcium binding causes a conformational change, allowing other proteins, the snare complex proteins, on both the vesicle and the terminal membrane to interact. Think of them zipping together.
Like molecular Velcro or zippers.
Kind of. They pull the vesicle membrane right up against the inside surface of the nerve terminal membrane, specifically at active zones.
And then?
They mediate the fusion of the two membranes. The vesicle essentially merges with the terminal membrane, pops open, and releases its contents, the neurotransmitter, into the synaptic cleft, that tiny gap between the cells.
That's exocytosis. The electrical signal caused calcium entry, which caused vesicle fusion and chemical release.
Exactly. It's a highly orchestrated process.
Yeah.
And it happens incredibly fast after the action potential arrives.
Okay, so chemical is now in the gap. Step 2.
Step 2. The neurotransmitter molecules diffuse across that very short distance of the synaptic cleft.
How short is it?
Tiny. Nanometers. So diffusion is quick. And on the other side, on the membrane of the next cell, the postsynaptic cell, are receptor proteins.
The locks for the neurotransmitter keys.
Perfect analogy. The transmitter binds specifically to its matching receptor proteins.
And what happens when the key goes in the lock?
That binding causes a change in the receptor, which in turn causes a change in the postsynaptic cell. Usually it changes the permeability of the postsynaptic membrane to certain ions.
Ah, so the chemical signal gets translated back into an electrical or ionic event on the other side.
Right. And there are two main ways this happens, depending on the receptor type.
Which are?
First, ligand-gated ion channels, also called ionotropic receptors. Here, the receptor is the ion channel. When the neurotransmitter binds, the channel opens directly.
So ions flow straight through?
Yep. If it's a channel for positive ions like sodium, they flow in, making the inside of the cell less negative, that's depolarization, and excitatory postsynaptic potential, or EPSP. If it's a channel for negative ions like chloride, they flow in, making it more negative, or stabilizing it, that's hyperpolarization, and inhibitory postsynaptic potential, or IPSP.
And this is fast.
Very fast. Direct coupling. Nicotinic HE receptors are a prime example. Also some glutamate GABA glycine receptors.
Okay, what's the second type?
G-protein coupled receptors, or GPCRs, also called metabotropic receptors. These are more indirect and generally slower.
How do they work?
When the transmitter binds, the receptor doesn't open a channel itself. Instead, it activates an intermediary molecule inside the cell called a G protein.
Okay.
This activated G protein then goes off and interacts with other proteins inside the cell. Maybe an enzyme that produces a second messenger chemical, or maybe it indirectly influences an ion channel nearby.
So it triggers a cascade of events inside the cell.
Exactly, a chain reaction. This allows for more complex signaling, amplification, and longer-lasting effects than the direct ion channels. Muscarinic AC receptors and adrenergic receptors for NEET PG are GPCRs.
Got it. Direct ion channels versus indirect G protein cascades. So step 3 must be what happens after the receptor is activated.
Right. Step 3, initiation of postsynaptic activity. If you get enough excitatory input, EPSPS, to push the postsynaptic membrane potential past its firing threshold, boom, it fires its own action potential. If it's a muscle cell, it contracts. If it's a gland cell, it secretes.
And if it's inhibitory input, IPSPS.
That makes it harder for the cell to reach threshold. It counteracts the excitation. The cell's overall response is basically the sum of all the EPSPs and IPSPs it's receiving at any given moment.
Okay, makes sense. But the signal can't just stay on forever, right? How does it get turned off?
Crucial point. Step 4, termination of the signal. The neurotransmitter has to be removed or inactivated very quickly from the synaptic cleft. Otherwise, the receptors would stay activated and you'd lose meaningful signaling.
How is it removed?
Depends on the transmitter. For acetylcholine, as we mentioned, there's that super efficient enzyme, acetylcholinesterase, ACE, sitting right there in the cleft.
For cleanup crew.
Exactly. It chops up ACE incredibly fast in less than a millisecond at the neuromuscular junction. Stops the signal cold.
What about norepinephrine? NE, no enzyme like that.
Not primarily in the cleft. For NE, the main termination mechanism is reuptake. Transporter proteins, specifically the norepinephrine transporter or NET, located on the membrane of the presynaptic nerve terminal that release the NE.
The one that sent the signal.
Yes. They actively pump the NE molecules back inside the terminal.
So it gets recycled.
Largely, yes. It's taken back up for repackaging into vesicles or breakdown inside the neuron. Some NE also diffuses away or gets taken up by other cells or broken down by enzymes elsewhere.
Yeah.
But that presynaptic reuptake via NET is the most important way its action is terminated at the synapse.
And other transmitters.
Amino acids like glutamate and GABA are also cleared mainly by transporter proteins on neurons and surrounding glial cells. Peptides are typically broken down by enzymes called peptidases in the cleft or on cell surfaces.
Okay, so removal is key. Is that the last step?
There's sort of a step 5, non-electrogenic functions. Besides these fast and off signals, neurotransmitters can also have slower, longer-term effects on the target cell.
Like what?
Influencing enzyme activity, changing the number or sensitivity of receptors over time, even affecting cell growth or survival. Trophic effects. It's not just about immediate excitation or inhibition.
Wow. And one more thing before we move to specific chemicals. The source mentioned modulation, fine-tuning the release itself.
Yes. Transmitter release isn't just on or off. The amount released per action potential can be adjusted, modulated, right at the presynaptic terminal.
Oh.
Through receptors located on the presynaptic terminal itself. There are 2 main types.
Auto and heteroreceptors?
Exactly. Autoreceptors respond to the neuron's own neurotransmitter. So when NE is released, some of it can bind back to alpha-2 adrenergic autoreceptors on the same terminal.
And what does that do?
Usually it acts as negative feedback. It tells the terminal, okay, there's enough NE out here now, maybe release a bit less next time. It helps regulate the signal strength. EGID does this too via M2, M4 muscarinic autoreceptors.
So a self-regulating mechanism. What about heteroreceptors?
Heteroreceptors on the terminal respond to transmitters released by other nearby neurons or cells.
So one nerve can influence another's release.
Precisely. This is really common where sympathetic and parasympathetic terminals lie close together. For example, NE, released from a sympathetic nerve, can bind to alpha-2 heteroreceptors on a nearby parasympathetic terminal and inhibit the release of ulsot from that terminal.
Ah, crosstalk between the systems.
Exactly. Allows for complex interactions and fine-tuning of organ function, adenosine, histamine, opioids. Lots of signals can modulate release via presynaptic heteroreceptors.
Okay, that's a fantastic breakdown of the whole junctional transmission process. Now, let's dive into the specifics, starting with the first big player. Acetylcholine, acetylate.
All right, ACE. The story starts with making it. It's synthesized right in the nerve terminal.
From what?
2 building blocks, choline and acetyl-CoA, an enzyme called choline acetyl transferase. Chakti puts them together.
And getting the choline into the nerve. That's important.
Very. It's often the rate-limiting step. There's a specific high-affinity choline transporter protein, CHT1, on the presynaptic membrane that actively pumps choline in from the synaptic cleft or extracellular fluid. Drugs called hemagglutiniums block this transporter, and that shuts down ACE synthesis.
So choline comes in, gets acetylated by chat to make ACE. Then what?
Then the ACE needs to be loaded into those synaptic vesicles for storage and later release.
Another transporter?
Yep, the vesicular ACE transporter, VACH. It sits on the vesicle membrane and uses the energy from a proton gradient, set up by a proton pump, also on the vesicle, to pump ACE from the cytoplasm into the vesicle. A drug called disamicol blocks this VACH.
So you need CHT1 to get choline in, chat to make ACE, and VACH to load it into vesicles.
Exactly. And these vesicles are packed thousands of ACE molecules per vesicle and many vesicles clustered at the terminal.
Release is then the calcium-triggered exocytosis we talked about involving snare proteins.
Correct. Action, potential, calcium influx, snares, immediate vesicle fusion, ACE released into the cleft.
And we know some toxins target this. botulinum toxin?
Yeah, Botox. It's incredibly potent. It works by specifically cleaving some of those essential snare proteins like synaptobrevin.
So the vesicles can't dock and fuse properly.
Right. It blocks ATG release, leading to muscle paralysis. That's why it's used cosmetically to smooth wrinkles, paralyzing small facial muscles, and medically for conditions involving muscle spasms or excessive gland secretion. Tetanus toxin works similarly, but affects inhibitory neurons more in the central nervous system.
Okay, so ATT is released, and then boom, acetylcholinesterase, ATV, destroys it in the cleft. Super fast in activation.
That's the primary fate at most synapses, especially the neuromuscular junction.
Mm-hmm.
Keeps the signal brief and precise.
Does ACE do anything else?
The source mentions some evidence for non-classical roles, maybe in synapse development or cell adhesion, but its main job is hydrolysis, breaking down AC.
Now, how does AC transmission differ depending on where it's happening? Skeletal muscle versus, say, the heart.
Big differences, mainly due to the receptors involved. At the neuromuscular junction, NMJ, the connection between a somatic motor nerve and skeletal muscle.
This is voluntary movement control.
Right, you have a very specialized synapse. AC is released and acts on nicotinic receptors, the muscle subtype, NM.
Those are the fast, direct ion channels?
Exactly. They let sodium ions rush into the muscle cell, causing a rapid depolarization called an end plate potential, EPP. If this EPP is big enough, it triggers an action potential in the muscle fiber, leading to contraction. Very direct, very fast, one-to-one signaling usually.
And you mentioned denervation supersensitivity earlier. If the nerve is cut,
the muscle fiber starts clustering extra nicotinic receptors all over its surface outside the original junction area. Makes it hypersensitive to any stray ASIA. Normally, innervation keeps those receptors tightly clustered right where they're needed.
Okay, that's skeletal muscle. What about autonomic targets? Smooth muscle, heart, glands?
Here, ASIA acts mainly on muscarinic receptors, M receptors.
And these are the slower GPCR types.
Correct. They work indirectly through those G protein signaling cascades inside the cell. The effect depends entirely on which M receptor subtype is present and what downstream pathways it links to in that specific cell type.
So, AC binding to an M receptor in the heart slows it down by increasing potassium permeability via M2 receptors, but AC binding to an M receptor in the gut smooth muscle might make it contract by increasing calcium via M3 receptors.
Precisely. It's modulating the intrinsic activity of the organ rather than necessarily initiating it like at the NMJ.
What about in the autonomic ganglia, that relay station?
They are the primary fast transmission from the preganglionic to the postganglionic neuron uses AC acting on nicotinic receptors again, the neuronal subtype NN.
So nicotinic again for fast excitation to pass the signal along the chain.
Exactly. Causes rapid depolarization of the postganglionic neuron. Though there can also be slower modulation in ganglia involving muscarinic receptors and other transmitters.
Okay, so nicotinic fast excitation, muscle, ganglia. Muscarinic slower modulation, organs, glands, effects vary by subtype.
That's the core distinction. And don't forget those presynaptic receptors we talked about, AC care release, can be inhibited by AC itself acting on M2M4 autoreceptors or by NE acting on alpha-2 heteroreceptors.
And the source mentioned non-neuronal AC2, cells outside the nervous system using it.
Yeah, surprisingly, many cells, immune cells, epithelial cells, can make AC and have cholinergic receptors. It seems to play roles in things like inflammation, cell proliferation. It's not just a neurotransmitter, a broader signaling molecule.
Fascinating. Okay, let's look closer at those two receptor families, nicotinic and muscarinic.
Diehl's original classification holds up well. Nicotinic receptors, NECARs, are stimulated by nicotine blocked by curare. Muscarinic receptors, MECRs, are stimulated by muscarine from certain mushrooms, blocked by atropine.
And the NCRs are the ligand-gated ion channels, made of 5 subunits.
Yes, permeable to positive ions like sodium and calcium, so they always cause excitation. Rapid response.
2 main types, muscle and neuronal.
Right. The muscle type, N, is at the NMJ, mediating contraction. Slightly different subunit makeup in fetal versus adult muscle. The neuronal type, N, is much more diverse, found in ganglia, adrenal medulla, and all over the CNS.
What do they do in the brain?
A lot. They are made of various combinations of different alpha and beta subunits. This creates many different subtypes with distinct properties and locations. They're found pre, post, and perisynaptically. Critically, many act as presynaptic auto or heteroreceptors.
Meaning they regulate the release of other neurotransmitter?
Exactly. Nicotinic receptors can enhance the release of AACE itself, but also dopamine, norepinephrine, glutamate, serotonin, GABA, depending on the receptor subtype and where it is. For instance, specific subtypes containing alpha-4 beta-2 or alpha-6 subunits are key regulators of dopamine release in reward pathways. Others, like alpha-7, influence glutamate transmission.
So they're major modulators of brain circuitry. That's complex.
Incredibly, their diversity allows for very fine-tuned control.
Okay, now the muscarinic receptors, MACHRs, these are the GPCRs.
Yes. Slower effects, mediated by G proteins and second messengers, can be excitatory or inhibitory. 5 subtypes, M1 through M5, encoded by different genes, found pretty much everywhere.
And they couple to different internal pathways.
Right. M1, M3, and M5 typically couple through G proteins called GQ11. This activates an enzyme, PLC, leading to increased intracellular calcium and activation of protein kinase C, PKC. Generally, these pathways are excitatory.
Like causing muscle contraction or gland secretion. M3 is big in smooth muscle.
Yep, M3 is dominant in many smooth muscles, like the bladder detrusor muscle and also glands. M1 is important in ganglia and gastric acid secretion. M5 is less widespread, found in places like the substantia nigra, involved in dopamine regulation.
Okay, what about M2 and M4?
They typically couple through inhibitory G proteins, GIGO. This pathway inhibits an enzyme called adenyl cyclase, decreasing levels of the second messenger CAMP. They also often activate certain potassium channels, leading to hyperpolarization, slowing things down, and inhibit calcium channels, so generally inhibitory effects.
Like M2 and the slowing heart rate.
Exactly. M2 is the predominant muscarinic receptor in the heart's pacemaker nodes. M4 is found more in the brain, involved in regulating movement and cognition, and acts as an important autoreceptor inhibiting AT release.
And most cells have more than one type.
Often, yes. The overall response depends on the mix of subtypes and the downstream signaling pathways present in that particular cell.
And these subtypes are important clinically.
Hugely. They are targets for drugs used to treat Alzheimer's, COPD, urinary incontinence, Parkinson's symptoms, motion sickness, and dysfunction is implicated in many diseases. Studying mice where specific receptor genes have been knocked out has been really revealing about their individual roles.
Okay, that's a deep dive into AA. Let's switch now to the other side of the ANS coin, adrenergic transmission. The language of the sympathetic system, norepinephrine, NE, and epinephrine, EPI.
Right, the catecholamines, along with dopamine, DA.
How are these made? It's a pathway, right?
Yes, a biosynthetic pathway, starting with the amino acid tyrosine.
Okay, first step.
Tyrosine is converted to dopa by an enzyme called tyrosine hydroxylase, TH. This is their crucial rate-limiting step. Its activity controls the overall production rate. It's regulated by nerve activity and feedback inhibition.
So TH is the bottleneck. Dopa becomes...
Dopa is converted to dopamine, DA, by another enzyme, aromatic L-amino acid decarboxylase, AAADC. This happens in the cytoplasm.
Okay, now we have dopamine. In most sympathetic nerves, what's next?
In nerves that release NE, dopamine is transported into the synaptic vesicles. Inside the vesicle, an enzyme called dopamine beta hydroxylase, DH, converts DA into norepinephrine, NE.
So NE synthesis actually finishes inside the storage vesicle.
Correct. But in cells that release adrenaline, like in the adrenal medulla.
There's one more step.
Yep. NE leaks out or is transported out of vesicles into the cytoplasm. There, an enzyme called phenylethanolamine N-methyltransferase, PNMT, adds a methyl group to NE, converting it into epinephrine, EPI, or adrenaline.
And then EPI goes back into vesicles.
Yes, it's transported back into storage vesicles using the same transporter that takes up dopamine.
So, tyrosine dopa, dopamine in vesicle, norepinephrine in cytoplasm, epinephrine, back into vesicle. Quite a journey.
It is. And deficiencies in these enzymes cause serious problems. TH deficiency leads to severe movement disorders. DH deficiency causes low blood pressure, droopy eyelids, shows how vital this pathway is.
So storage is in vesicles again, using a transporter.
Yeah, catecholamines, DA, NE, EPI, and also serotonin are pumped from the cytoplasm into vesicles by the vesicular monoamine transporter 2, VMAT2. This uses a proton gradient similar to VHE for HEI.
And drugs can block this. Reserpine.
Reserpine is a classic VMAT blocker. It prevents storage, so the transmitters get broken down in the cytoplasm, leading to depletion. Used to be used for hypertension, but has significant side effects.
And like HE vesicles, these adrenergic vesicles often contain other stuff too.
Yes, very commonly. AD is often co-stored and co-released with ATP and the peptide neuropeptide Y, NPY.
More co-transmission. How do ATP and NPY get in?
NPY is a peptide. So it's made via the usual protein synthesis route and packaged into large, dense core vesicles. ATP is transported into vesicles by a specific vesicular nucleotide transporter, VNUT.
And when released, they act on their own receptors.
Yep, ADP acts on P2X ion channels and P2Y GPCRs, purinergic receptors. NPY acts on NPY receptors, which are GPCRs. They add further layers to the sympathetic signal. ATP provides a fast excitatory component, while NPY often causes slower, longer-lasting effects, like vasoconstriction.
Okay, release is again calcium-dependent exocytosis with snares.
Same fundamental mechanism as for HET release.
Now, termination. We said for NE, it's mainly reuptake back into the nerve terminal.
That's the major route for clearing NE released from sympathetic nerves. The norepinephrine transporter, NET, also called uptake 1, is the key player here.
What is NET exactly?
It's a protein embedded in the presynaptic nerve membrane. It uses the sodium gradient to actively pump NE, and to a lesser extent EPI, from the synaptic cleft back into the neuron cytoplasm.
And this is where drugs like cocaine and tricyclics work.
Exactly. Cocaine, tricyclic, antidepressants like metformin, they block NET. This prevents NE reuptake, so NE stays in the synaptic cleft longer, enhancing sympathetic signaling.
Is there any other uptake?
Yes. There's also uptake 2, mediated by transporters like ENT or OCT3, on non-neuronal cells, like smooth muscle, glands, glial cells. This transporter has a lower affinity for NE, but a higher capacity. It's more important for clearing circulating catecholamines, like adrenaline released from the adrenal gland, or for handling higher concentrations. But for neuronally released NE, NET, uptake 1, is responsible for clearing the vast majority, maybe close to 90%.
So NET function is really critical for normal sympathetic control.
Absolutely. The source mentions NET knockout mice show major changes in heart function and behavior. And human mutations in the NET gene are linked to things like orthostatic intolerance, problems maintaining blood pressure when standing up, or potentially mood disorders. It really highlights the transporter's importance.
And some drugs work indirectly, by getting into the nerve via NET.
Yeah, drugs like tyramine, found in aged cheeses, red wine, or ephedrine, they are taken up by NET into the nerve terminal. Once inside, they displace NE from the storage vesicles, causing NE to leak out into the synapse through NET working in reverse. They're indirect sympathomimetics. They cause NE release without nerve firing.
Clever. Okay, so NEPI released, signal is mainly terminated by NET reuptake. What do they bind to on the postsynaptic side?
They bind to adrenergic receptors. These are all GPCRs, and they are broadly divided into 2 main families, alpha aviary-eupilzilla receptors.
And there are subtypes within those.
Yes, alpha 1, alpha 2, beta 1, beta 2, beta 3. Each subtype couples to different G proteins and intracellular pathways, leading to different effects in different tissues.
Like beta 1 receptors, mainly in the heart increasing heart rate, beta 2 in the lungs causing bronchodilation.
Exactly. Alpha 1 often causes smooth muscle contraction, like in blood vessels. Alpha 2, as we mentioned, is often found presynaptically as an autoreceptor that inhibits NE release, but also postsynaptically in some tissues. It's the specific combination of receptor subtypes on a target organ that determines its overall response to sympathetic stimulation.
So the complexity comes from both the co-transmitters and the variety of receptor subtypes.
Precisely.
Now, AC and NEPI are the big players, but the source briefly mentioned others involved in the NANC transmitters.
Right. Non-adrenergic, non-cholinergic. Acknowledging that not all autonomic transmission uses the classical duo. Purine's ATP and its breakdown product adenosine are important examples.
ATP is released with NE and AC sometimes?
Often, yes. And it acts on its own P2 receptors. Adenosine acts on P1 receptors. They play roles in blood flow regulation, gut motility, bladder function, pain signaling.
And nitric oxide, NO.
Yes. NO is a key NNC transmitter, particularly in inhibitory pathways in the gut, relaxing smooth muscle, and mediating vasodilation caused by some parasympathetic nerves. And related signaling molecules like endothelin, produced by blood vessel lining cells, also heavily modulate vascular tone. It's a busy chemical landscape.
Okay, wow. We've covered a lot of ground. From the big map, somatic versus autonomic, sympathetic versus parasympathetic, right down to the molecules, the synthesis, storage, release, receptors, and removal of SE and NE.
It's a journey from macroscopic organization down to nanoscale molecular machines working at the synapse.
And the key takeaway seems to be this whole process of neurotransmission. It's incredibly elegant, but also tightly controlled at every single step.
Absolutely. Making the messenger, packaging it, releasing it precisely with an electrical trigger, having it find the right receptor lock, causing a specific effect, and then cleaning it up immediately so the system is ready for the next signal. It's biochemical communication at its finest.
And really understanding these steps, these molecules, it's not just academic, is it? It's fundamental to medicine.
Completely. So many drugs work, by targeting these specific mechanisms, drugs that block synthesis or storage, like razorpine, or release, like Botox, or reuptake, like antidepressants and cocaine targeting NET or serotonin transporters, or drugs that mimic the natural transmitter agonists, or block the receptors antagonists for HCRE or NEET PG.
Treating everything from high blood pressure, asthma, allergies, gut disorders, depression, anxiety, Parkinson's, the list goes on.
It all comes back to manipulating this chemical signaling in specific pathways. And of course, many toxins and diseases also disrupt these same processes, highlighting their fragility too.
So thinking about the sheer scale, billions of neurons, trillions of synapses, this constant symphony of chemical signals using HCANE, dopamine, serotonin, glutamate, GABA, peptides, ATP, NO, all interacting, modulating each other, activating specific receptor subtypes.
Yeah, it's mind-bogglingly complex when you really start to think about it. The level of coordination required moment to moment just to keep you functioning, breathing, thinking, feeling, reacting appropriately to your environment.
It really makes you wonder, considering that vast complexity and all those moving parts, what happens when just one small part of that intricate chemical machinery, just one type of receptor or transporter, is slightly out of tune? How many subtle or not so subtle effects could that cascade into throughout the whole system?
That's the question, isn't it? And something researchers are constantly exploring.